All right, how's it going, everyone? Welcome back. Hope you're all doing well. And this is going to start our first lecture in our next module. And this lecture is called Defining Evolution. So we're going to get into the very basics and the beginnings of how scientists started to think about organisms changing over time and how they thought about, reasons why you might look like your parents and organisms look like, their parental organisms. So, and then at the very end, we'll talk about the first part of evolution. So let's just get right into it. First, I want to introduce you to a man named Gregory Mendel and his world famous by now pea experiments. He was an Austrian monk, so not Australian, as some students will like to put on their exams, but he was an Austrian monk. And he does this seminal work, and it was accomplished using the garden pea, Pisum sativum, and he does this to study inheritance. Now, Gregor Mendel, being an Austrian monk, had a lot of free time on his hands, right? He wasn't going out and partying, he wasn't going out and dating. He had a lot of free time. And so what he did in that free time was he basically grew a lot of plants, and he took meticulous notes and data on how these plants worked. The pea plant that he studied are described as true breeding. And what that means is that the plants will always produce offspring that look like the parent. And so there's a number of different traits about these plants. Some of them are tall, some of them are short. Some of them have purple flowers, some of them have white flowers. Some of them have green peas, some of them have yellow peas. Some of them have like smooth surfaced peas, and some of them are shriveled peas. But what you always found is that if you made babies of two purple flowered plants, their babies would have purple flowers. If you had two babies with white flowered plants, they would have white flowers, and so on and so forth. So they were called true breeding. The other thing about the Mendel's garden peas that he used were that they grow quickly. Okay, he could produce millions of plants in his lifetime. And that's important because when we talk about genetics and we talk about inheritance, a lot of these things can happen due to chance. And so you have to have large numbers of your results in order to prove that something is going to happen not due to chance, okay? To show that there are actually patterns in your data. And that's important in any science, is to have lots and lots of copious amounts of data. So if he were trying to study inheritance in humans, well, he would only be able to go through a couple generations before he was no longer with us. Whereas if you take another organism like this garden pea that can produce many, 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 many times in a human's lifetime, that obviously makes a stronger case. Mendel did what we call hybridizations, which involves taking two true breeding individuals, but they have different traits. Okay, so maybe you take purple flowers and white flowers and make them have babies and see what happens. Plants used in the first generation were called the pea, or the parental generation. Okay, so it's basically like your parents. They're the pea generation, okay? Mendel collected the seeds produced by the pea plants that resulted from each cross and grew them the following season. Those offspring were called the F1, or the first filial. Filial is daughter or son in Latin. And so the F1 generation, you could think of that as you. Your parents are the P generation. They had an F1 generation, okay? And then if you have children, they'll be the F2, the second filial generation, okay? That's just how that works. P generation are the parents. F1 are the sons and daughters, or, if we're talking about animals with sexes, but in terms of plants, just the next generation. Garden pea characteristics are going to help Mendel reveal the basics of heredity. He was able to show that crossing true breeding plants with true breeding plants always will result in the same trait. Let's talk about flower color. Mendel applied the pollen from a plant with purple flowers to the stigma of a plant with white flowers. and he found that 100% of the F1, or basically all of the children, hybrid generation, had purple flowers. So if you mate purple flowers with white flowers, their babies all have purple flowers. Absolutely none of them will have the white flower. It just disappears completely. But importantly, Mendel doesn't stop there. He doesn't stop and go, oh, well, I guess that's it, right? There's more questions that he asks. So what he does is he takes two of the babies from that F1 generation that are going to have purple flowers, and he mates them. Now, this is not disgusting because we're talking about flowers. You obviously can't do this when we talk about humans, correct? But if you take two of the F1 generations, two of the children from the purple and white flowers that began to have all purple flowers, he does this over and over, and he finds that 705 plants in the F2 generation, which is the next generation, had purple flowers, but 224 had white flowers. So those white flowers reappear. And think about that. You're taking two individuals with purple flowers, and suddenly, in a 3-to-1 ratio, they're going to have white flowers. So that's very interesting because suddenly we find out that, hey, that somehow these purple, they show up purple, they have the ability to make children that have white flowers, to make offspring. They're not their children, they have offspring that have white flowers. That's really, really interesting. He concludes that the characteristics could be divided into what he called expressed and latent traits. And basically he calls them dominant and recessive traits. Dominant traits are those that are inherited unchanged in a hybridization. So the purple flower, the purple and the white in the P generation made purple flowers. So the purple color must be dominant. Recessive traits become latent, or what that means is to disappear in the offspring of the first hybridization. So the white flowers in the F1 generation are totally gone. 100% of them were all purple, so that is the recessive or the latent trait. Here's what that looks like. So here's our P generation. So remember, these violet or purple flowers, if you took two of them, they would make all purple. If you took two whites, they would make all white. But if you cross a purple and a white, 100% of your F1 generation becomes purple. And so he didn't stop there. He said, okay, what if I make two of these have babies? Well, in a 3-to-1 ratio, he finds that they make purple flowers and one white flower. So for every 3 purple, you'll get a white. And it was like 705 to 224, which is a 3-to-1 ratio. And what he concludes from this is that, well, since the purple is always there and the white disappears in this first hybridization, purple is dominant, white is recessive. So this brings up phenotype and genotype, two words that you're going to have to get to know. So gene variants are called alleles, and you get one copy from each parent. Two alleles for a given gene are expressed and interact to produce the physical characteristics. The observable traits that are expressed by an organism are referred to as its phenotype. So this flower is purple, it has a purple phenotype. This flower is white, its phenotype is white, okay? All organisms' underlying genetic makeup consisting both of the physically visible and the non-expressed alleles is called its genotype. So, what I'm going to show you in a second is that purple flowers, what you see, could have one genotype or another genotype. Basically, the genotype is what your A's, T's, G's, and C's are. It's your genetic makeup, okay? But what you're going to find out in just a second is that your genetic makeup could be different, but you could look phenotypically. The phenotype, the way that you look, could be identical, okay? That's what we're going to show in just a second. So Mendel's hybridization experiments demonstrate the difference between phenotype and genotype. So he also does one with seed color. And what he finds, and he doesn't know any of these words, genes, alleles, he doesn't know any, he doesn't have the ability to look through a microscope and figure out, oh, there's genetic information. This is happening way, way before we ever find this out. But basically, what we now know is that seed color is governed by a single gene with two alleles. The yellow seed allele, which is dominant, and the green seed allele, which is recessive. When true breeding plants were cross-fertilized, in which one parent had yellow seeds and one had green seeds, all of the F1 hybrids had yellow seeds. So think about that in your head for just a second. Okay, so if I took the true breeding yellows and the true breeding greens, and in the F1 they all had yellow, which one is the dominant, which one is the recessive? So hopefully you should figure out that the yellow is dominant because they all had yellow in the F1. Green's going to be recessive. The hybrid offspring were phenotypically identical to the true breeding parent with the yellow seeds, right? They look identical. They have yellow seeds, and they don't look any different at all. If I put them all in a bucket, put a bunch of yellow peas in a bucket, and I said which one were from the parent generation, which one for the F1, you wouldn't be able to tell. However, we know that the allele donated by the parent with the green seeds was not simply lost, because if you cross two of those F1s and the F2, you get the green seed reappearing. So therefore, the F1 plants must have been genotypically different from the parent with yellow seeds. I know you're probably freaking out right now, but here's what this looks like. So we had the true breeding yellow and the true breeding green, and we do a cross in the P generation, and we get all yellows. What that means is yellow is dominant, green is recessive. Okay, I think we should probably are pretty good on that. But we also know that green is not just gone forever, because if we cross two of these, we get in a 3-to-1 ratio the green returning. And so although phenotypically what they look like are yellow, genotypically they're different. They have to be different, because if you cross two of these, you're only ever going to get yellow. But if you cross two of these, you do get green. And here's what that looks like. So when we talk about genotypes, we usually take a letter for the color or how tall it is, whatever trait it is, we ascribe it a letter. So for this, we got yellow and green, and they decided to use a capital Y for the dominant, a lowercase y for the recessive. So you always use a capital for the dominant, a lowercase for the recessive. Okay? And so if you look at this, you can think of this as the sex chromosomes. Okay? So each parent, mom and dad, or dad and mom, whichever one you want to say is mom or dad, gives one up. You can only give up one. And in this case, the yellow can only give up a dominant yellow, because whenever you cross two of these yellows, they only make yellow. So it's got to give up a dominant yellow. The lowercase y, makes green. And so if you cross two of these greens, you can only get green. Okay? So it can only give up little y. It's got two copies because it has to get one from its mom and its dad, back in the grandparents, right? Now, when these cross, they can only give up one. Well, this can only give up big Y, and this can only give up little y, leading to big Y, little y being the genotype for this generation. Its phenotype is yellow. Its genotype is what we call heterozygous. Heterozygous meaning that it has one copy of the big allele, dominant allele, and one copy of the little allele. The parents were what we call homozygous. Homo being the same, right? Homozygous, having the same allele for each. Homozygous dominant, they have two dominants, or homozygous recessive. So here we have a heterozygous, meaning like heterosexual, male and female, but that just means there's two different types. Heterozygous, big Y, little y, dominant recessive. Yellow is what they look like phenotypically. Genotypically, they're heterozygous, whereas this one, genotypically, is homozygous dominant, whereas phenotypically, it's yellow. Just like this is phenotypically yellow, but they are different genotypically. Okay? Now, when we cross two of these heterozygous yellow organisms, it can either give up a big Y, and then let's say there was another one here, and a big Y. It could give up a big Y, and the other one could give up a little y. Let's say there's another one over here. You see that? It could give up that. Or it could give up two little y's. Okay? And it happens so happens to work out that if you only get one copy of the dominant allele, you present the dominant trait in your phenotype. And so since you can get two big Y's, or you could just get one big Y while the other one gives up a little y, 75% of the offspring are going to be yellow, and only 1/4 of the time, or 25%, will you get both of these parents giving up a little y to make green. So hopefully that makes sense between phenotype and genotype. Of course, we'll talk about this more and obviously ask me questions if you have any, but we'll keep moving. So like we just said, diploid organisms like us, like humans, that have a copy from mom and a copy from dad of the same gene, they are homozygous for a gene if they have two identical alleles for the same trait. You could be homozygous dominant if you have two of the dominant alleles, two of the capital Ys in that example, or homozygous recessive if you have two recessive alleles, the two little y's in that example. Diploid organisms that are heterozygous for a gene, like I said, have two different alleles. They have the dominant and they have the recessive. So they're heterozygous. Notice how we don't have to say heterozygous dominant or heterozygous recessive, because heterozygous just inherently means having both. Homozygous, you can't say homozygous and leave it at that, because you have to tell me, tell anyone, if they have two copies of the dominant or two copies of the recessive allele. What are alleles? Alleles are mutations. Well, mutations are the source of new alleles in a population. The different forms of a gene are called the alleles. And here's an example that's actually not really the case, but it just helps to illustrate it. The gene that controls eye color will have different alleles for blue, green, or brown, right? And depending on how these two interact, let's say mom has blue, dad's got brown, blue and green, two blues, two greens, two browns, you can only give up whatever alleles you have, okay? And that's not really how it works with eye color. People are always curious about that. It's a little more different. It's a little more complicated than just having two copies of an allele for the eye color. But anyway, all that this is getting at is alleles are different forms of the same gene. So you have a gene for flower color, and there were two different alleles, purple or white, okay? That's basically getting at that. Now, what's evolution? Evolution is defined as the change in allele frequency over time in a population. Allele frequencies are always going to remain the same in a population unless one of these four things happens: natural selection, mutation, genetic drift, or gene flow. Okay, and we're going to talk about all four of these as we continue on in this lecture series. But to just point out that that's all evolution is: how frequently an allele pops up in a population. Okay, let's say you had a population of all humans that had all brown eyes. Well, if there's no blue eyes, there's no blue eyes, right? So how could a blue-eyed organism suddenly get in? Well, one of these things has to happen. The easiest one that you probably would think of is some sort of mutation, right? Where two brown-eyed individuals have a child, but that child has a mutation in its genome that produces blue eyes. That's one example. We'll talk about the rest now. So the first one, and the only one we're going to do today, is called gene flow. Gene flow is the flow of alleles in and out of a population due to migration. Genes can travel through individuals or gametes. Alleles introduced may be common in the source population, but rare in the new population. So remember our blue eye, brown eye example I just gave? Well, let's just switch it to green beetles and brown beetles. So we've got a population of all brown and all green beetles, and they're far away from each other, so that they're not commonly mixing. But some sort of immigration or emigration from here or immigration to here happens, where all of a sudden this brown individual meets up with this green population. Now, notice there were no brown alleles in this population, but if this individual comes in and mates, there's going to get an influx of this brown allele. And that is, by definition, evolution, because we have changed the frequency of alleles in this population. This population will now be able to evolve because it has the brown alleles. So that's the first example of how evolution can happen. It's gene flow, where either individuals or gametes get introduced that carry alleles that previously were not present in the population. Like I said, immigration, individuals join a population, changing allele frequencies, or emigration, individuals leave a population. Let's say this was a blue one, and he was the only blue individual in this small population, and he left, well, there would be no more blue alleles in that brown population, and so that would just lead to that population also evolving away from the blue. So here are some examples of gene flow. A population of flowers on one side of the river transports pollen to the flowers on the other side of the river, producing offspring. Blue-eyed people from Sweden move to a small town in Mexico where people all have brown eyes. When they mate, some of their children now have blue eyes. Some birds with shorter beaks enter a population of birds with much longer beaks, resulting in the hatching of birds with in-between beak size. A Maine Coon cat is brought to an island where only tabby cats live. After mating with other cats on the island, some of the kittens have bushy tails and tufted ears. Rhinos from one herd move to a new area and breed with rhinos of a completely different herd. Pollen from trees is blown far, far away to a completely separate group of trees and pollinate their flowers, producing trees with genetic characteristics of each population. Several red foxes move into and mate with a silver fox population. Red parrots are brought on an expedition to a remote section of jungle with only blue parrots, introducing color variation into the gene pool of jungle parrots. Tigers with enhanced sensitivity in the dark mate with a group of tigers with less sensitive eyes, allowing a greater population of tigers with enhanced eyesight to be born after a few generations. So this is one example of how evolution can occur. Gene flow, the movement of alleles, new alleles into and out of a population. So hopefully that one all made sense. Let me know if there's any questions on it, guys, and I will see you in the next one very shortly.